Hey guys, welcome back. This is Lei. In this video, I'm going to show you how to analyze CUT&RUN or CUT&Tag data. So what is CUT&RUN or CUT&Tag? Like ChIP-seq, they are technologies used to study DNA-protein interactions. They also require specific antibody binding. However, unlike ChIP-seq, CUT&RUN and CUT&Tag don't require cross-linking and random fragmentation. They start with live cells. In CUT&RUN, the first step is antibody protein of interest binding. Next, an enzyme called PA-amylase binds to the antibody. Upon activation by the calcium ions, this enzyme can precisely cut flanking regions of the protein binding sites. These regions are then subjected to sequencing. CUT&Tag follow the same process, except the enzyme used is called PATN5. This enzyme is activated by magnesium ions, and also it can insert adapters at the same time when it cuts. And then the resulted regions are subjected to sequencing, just like in CUT&RUN. Compared to ChIP-seq, what advantages does CUT&RUN or CUT&Tag offer? Well, CUT&RUN or CUT&Tag needs a lot less cells. The background signal is very low, resolution excellent, processing time low, cost low. So basically everything is better in CUT&RUN and Tag than ChIP-seq. But when do we need CUT&RUN and when do we need CUT&Tag? If you need your data to be clean and precise, and your focus is on transcription factor, CUT&RUN is your choice. If your study is focused on histone marker modifications, and you need your data to be faster and cheaper, CUT&Tag is your choice. The workflow for processing CUT&RUN or CUT&Tag data is exactly the same as the one we use for ChIP-seq data analysis, maybe with a few tweaks. To refresh your memory, let me quickly walk you through this workflow again. First step, set up the environment. On a powerful Linux system, let's first activate our conda environment we created for ChIP-seq data analysis in our previous tutorial. You can install a new peak calling tool, SEACR, for CUT&RUN or CUT&Tag, but you don't have to. MACS2 works just fine. We download our example data from GO public dataset, rename the downloaded FASTQ files with some meaningful names. We trim the adapters using our old friend Trim Galore. You should be super familiar with this tool if you have followed my tutorials. We assign long directory names or file names to some variables to keep our code concise. We use BWA-MEM to map our reads against our reference genome. Pay attention to the input and output of the command line too. You don't need this line. After alignment, we have to go through some quality control steps. First, convert the resulted SAM files to BAM file. Sort the BAM file, and remove duplicates using PyCard mark duplicates. And then we filter out low quality reads with Sambaba and better tools. And don't forget to index the resulted BAM file. Next is the specific for CUT&RUN and CUT&Tag to remove the mitochondrial alignments. This line means only keep the standard chromosomes 1 to 22 and XY, and we don't need chromatin mitochondria. For peak calling, MACS2 works just fine. Pay attention to the input and output of this command line tool and other parameters as I demonstrated before. This is the output for MACS2. If you want to call the peaks with SEACR, you have to go the extra mile. We have to convert BAM files into batgraph files because they are the inputs SEACR accepts. Here, we do the conversion for both the experiment sample and the control sample. And then we sort the converted bad graph files for both samples. Now we have the bad graph files, we can call the peaks using SEACR. Input the bad graph files for both samples. Normalization against control. Relaxed so we can filter afterwards. Output folder over here. You can also call the peaks without control sample, but just don't do it. And this is the format of the output of SEACR. For peak annotation, we can use annotated peaks.pl function from the homework package. The peak file from MACS2 and the peak file from SEACR can be directly used as the inputs of this command line too. This is the format of the resulted annotated peak file. Now you can directly associate genes with the peaks. For peak visualization and motif analysis, you can use the same tools I demonstrated in my previous ChIP-seq tutorial. They are Deep Tools, Homework Package, and IGV. This is all I have for today's tutorial. Subscribe if you like it, and I will see you in the next video.